Hello everyone, I just want to deal with all of this today, okay? So now I'm going to be looking at another part of the XN line, which is called, I'm going to be focusing on TS 500, but they all work with the same principle. Another type of TS, but basically means tube sorter. T means tube, S means sorter, then 500 is what I'm going to be looking at, okay? When you talk about a tube sorter, remember what I said before. I said, once from the track, the sample is loaded, it goes to the full block count, okay? Analyzer, that will run the full block count. After that, if any of the sample require block film, it will go to the SP10 for the film to be made. Then after that, then the next thing, it will be transported to the tube sorter. Then that is where it can sort it, okay? I said tube sorter means it will scan the barcode of that sample, and from once it scans the barcode, it can now check everything about this sample. Has it been done? So it will scan the barcode, then following the scanning of the barcode, the tube sorter will then check, has everything been done? Remember, let me give you an example of this. Let's say for some times, there's a mislabeling. Something that is for full block count has been labeled for biochemistry test, and someone has loaded it on the analyzer. It will still go through. Then when you go to the tube sorter will scan the barcode and say, oh, I don't know this. It's going to sort it by putting it on any area, because we'll label under the tube sorter, we'll label each areas. So under the tube sorter, you can see archive rack. The archive rack is where it can put it in if all the tests have been done. Then you have the ESR rack, which we'll call the buffer rack. So once any sample that is left for ESR, it will put it there. Then you also have other aspects that can show you, maybe I don't know what this test is, or maybe there's still something outstanding and something like that. So something like that biochemistry sample label that is on that full block count sample, once it scans it doesn't know what it is. It's going to put it like, I don't know what this is. Also, something like maybe, like when the label is not properly labeled, maybe the label has worn off, it's not very clear. The analyzer is not able to scan the barcode properly, so it's not able to detect what the test is all about. So again, when it comes to the tube sorter will scan it all. I don't know what this is, okay? Then sometimes it will also scan the barcode depending, and maybe it has done the full block count, but it wants you to repeat it. You know what I was talking about, WPC, okay? So maybe it has done it and it has flagged blast or abnormal lymphocyte or something like that. Maybe the analyzer think that you should repeat this using WPC channel. Then the tube sorter will scan that barcode. Once it scans it, will say, even though it has done the full block count, but it is not finished. So I want you to repeat it. It's going to put it on a standing aspect of the rack, okay, of that tube sorter, okay? It will put it on a standing space or area. Then, so that is how it works. Then when you scan a barcode of the one that is completed, it will just archive it. So that's the difference between archive. So when you talk about archive, it means that everything is done. So these other ones of sorting it means putting it into where it can suggest what is remaining. Is it that I don't know what this sample is, or there is something outstanding, or it requires ESR? Now when you put all the samples on the ESR, what happens then, whenever it is ready for the ESR to be done, it can then be transported to the ESR analyzer, which is the next video I'm going to make, which is called interliner. Then that will then run the ESR there. Does that make sense? And once the ESR finished and you come and put it back on the tube sorter, once it scanned it said everything is done, it will just archive it for you. Okay, so now let's get into it. So when you talk about the archive, so basically I've just summarize what this tube sorter means. So now once the full block count sample has been done from the analyzer, anyone required for film will go to the film. After the film, it will then go to the tube sorter. So that is what the tube sorter is doing. It's scanning the barcode of any sample that comes in there to determine, okay, do I need to do anything in terms of, do I need to archive it like everything is done, or do I need to sort it like, I don't know what it is, unidentified, I don't know what it is, please could you check this, or maybe something is still outstanding, something like that, okay? So that is what tube sorter on its own does. That's why with the tube sorter, you can have something like unknown, outstanding, and of course you have buffer rack, and of course then you have archive rack, okay? So that is how the tube sorter work. Another thing I'm going to tell you about the tube sorter, because I'm trying to give many of you guys idea, to have an idea of what goes on in the lab. So that's why I try to explain this in detail. So now that the tube sorter have done something like archive, finish, it has also said unknown, I don't know what this sample is, it put it on unknown area, okay? Or maybe buffer rack, this is for ESR, you put it there, or maybe there's still something outstanding, it will put it there. Now, what I'm going to tell you, with the tube sorter, it will be able to know, once you check your what we call extended processing unit, which is what we call EPU on the Sysmex, is a Sysmex laboratory information management system. So once you look at that, it can actually tell you the sample, the particular sample rack it is. Is it now? In fact, when you check on that very EPU, you will also see, with the help of the tube sorter, you can also see, whether that has been archived. If the sample has not gotten to the tube sorter, you will also know. Anyway, let me focus on the tube sorter. So, sometimes you might be looking for samples, or maybe you processed the food block and initially they requested for food block, and after some time, the sample has been archived as finished, but the doctor requested for ESR. So once the doctor requested for erythrocyte segmentation rate, which is ESR, what are you going to do then? Then you now have to look at the patient detail, look it up onto the laboratory information management system. Then from there you can now check the lab number of that very food blocker sample of the patient. Then check it under the EPU. When you check it under the EPU, it will show you TS. So once you click the TS, it will then show you the position where it is on the rack inside the TS. For example, it will show you that the sample is on rack 1, position this, or rack 2, position this. What it means then is that there is aspect of the tube sorter we call initialization. So I'm going to start all over in terms of initialization so that you know how that functions. So once we initialize that tube sorter, what it means that you clear off any memory in terms of maybe, because sometimes in labs, some people may have something like maybe rack 1 to rack 50, as the case may be. So once you start storing up to rack 50, you need another rack 1. Is it now? So you can't continue with rack 51, rack 50, because you don't have 51, you don't have 52. And that tube sorter does not know that. So the tube sorter will continue to count 1, 2, 3, 4, up to whatever. So you know your last rack. So once the tube sorter has archived the last rack, what you then need to do is to initialize it. When you initialize it, you are trying to say start all over again. Okay, so then you can now discard all the maybe samples in rack 1, rack 2, rack 3, up to how many that you want to discard. Then you can now put back, once you initialize it, you then need to put back rack 1, okay, and put back rack 2. So some of them, depending on the tube sorter, might have up to, for you to archive for rack 1, rack 2, rack 3, as the case may be. So, but let's say rack 1, so you put rack 1, you put rack 2, or you put rack, depending, some places will arrange it like this: rack 1A, rack 1B. So you just put rack 1A, rack 1B, okay? You put it there. So once you've initialized it, what the tube sorter will then do, once another sample is scanned, it now know that from now I'm going to start counting 1. Then it will count 1 and then start storing it. That way when you check, you now see that those ones will now become 1 or 2, depending on the case maybe. But another thing I'm going to tell you is for you to know that each of the rack of the tube sorter can store about 125. So when you have each of the rack of the archived rack will store about 125 samples. So meaning when you have 1A and 1B, you are looking at 125 plus 125, you're looking at 250. Okay, so that's what happened. Is that okay? So now with that, once it's stored from 1, 2, 3, 4 up to 125, it now know that the next thing I'm going to store, it will now record it as, because if the first rack end in 125, okay, 125, that means the second rack will start with what? 126. So when you see 126, you now know you should be looking at rack 1B, not rack 1A, because each of the rack stores 125. So if you have rack 1A, depending on how you program it, how they program it in the lab. Some people program it 1, 2, 3, 4, as the case may be. Okay. Anyway, so if you have it as rack 1A, so it will store the rack 1A up to 125. Then after that, the rack 1B, once you see 126, you should not look for that sample on rack 1A, because rack 1A will finish at 125. So 126, you start looking at rack 1B and so on. So that is how that happened with the tube sorter. So it will sort all of those things out and store it for you to easily to be able to go and pick up the sample. Because sometimes, even biochemistry might come to you and say, oh, have you finished your food blocker? Can I use your sample for HbA1c or something like that? So once such a thing happens, it is easy for you to just take note the number of the sample and check it on the EPU. If you click the tube sorter, which is TS, then you can be able to see the position of that very sample. So it helps you to basically quickly, go and get the sample and do any other thing that you want to do for the sample. It could also be because it has archived it and you want to make a blood film. The analyzer thinks that it shouldn't make a film, but you think you should make a film as a BMS. Then, because it has been archived, once you click on the TS, it will also help you to be able to go and pick the sample, wherever it is, because you will see it there another. Why this tube sorter is very, very important is because then, if you can't see any sample, and you look at the history, it has not gone on the tube sorter. Sometimes sample may drop off on the line before coming to the tube sorter. Okay, so then you can check it. Did it drop off anywhere or something like that? So anyway, that's how the tube sorter works. So again, the tube sorter will help to sort the sample to know whether is it unknown, is it outstanding, is it finished in terms of archive, or does it require ESR. So here you go. That's your tube sorter. If you have any question, please put it in the comment section. And of course, let me know how this video has helped you, or maybe how interesting you find the video. Can I also ask you to please subscribe, like, and share. Thank you very much till I come back away again. Bye-bye.